Today, we are going to talk about device-based approaches for oral delivery of biologics. We will conclude by introducing the JetCAP technology in more detail and comparing the various systems. Challenges of oral delivery of macromolecules is quite significant. Charged molecules larger than 1,000 grams per mole have very low bioavailability and are often not measurable. Despite many decades of trying, oral bioavailability from formulation approaches is less than 2% for peptides and much lower for proteins. In the last 10 years or so, some very promising work using microneedle systems in the GI has been obtained. In this presentation, device approaches to macromolecule delivery will be discussed, and the JetCAP technology will be discussed in more detail. The reference at the bottom of this slide is a good recent high-level summary of oral biologics delivery with some key references in it as well. It is fairly obvious why we might want to deliver biologics orally. Patient convenience is paramount, and oral biologics, if achievable, would open up the broader patient population to a whole set of drugs only deliverable currently by injection. From a pharma perspective and a delivery company perspective, the top biologics represent $60 billion in sales in 2018, and this number could be much larger if oral delivery systems were achievable. In addition, the number of biologics approved by the FDA, as shown in the panel on the right, is increasing in proportion over the last 6 years. So it's fairly obvious why this would be an interest. We're going to talk about device approaches to oral delivery. And recently, there have been a number of approaches that look interesting. There are really two main categories for oral delivery using devices. One is the microneedle-based systems, and the other are liquid-based delivery systems. Rani is the most advanced system with phase 1 data published earlier this year in a press release. Progenity technology has been in the clinic with no data reported from their trials as yet. And two systems are under evaluation by the MIT group, along with Novo support, and they've presented preclinical data. We'll discuss JetCAP technology in more detail, and there are two other device-based approaches which are being evaluated for peptide delivery, and Trega will be discussed briefly, and the Linder technology may or may not be suitable for biologics. Rani Therapeutics was founded by my friend Mir Imran, who introduced me to the system about 10 years ago when all he had was mini pig PK data for several biologics, including insulin and human growth hormone. They've recently reported bioavailability in humans for octreotide of 70%. The system is based on a microneedle coated with drug, which is pushed into the wall of the GI with a bellows inflatable system, which also exposes the microneedle. Drug-coated microneedles typically can achieve a drug loading of 1 to 3 milligrams of active drug. One big advantage of this system is that it is an enteric-coated capsule which dissolves into small, malleable pieces for excretion. I'm very thankful to Mir for introducing me to this revolutionary way of delivering biologics using a device-based system. The Soma device from the MIT group is an ingenious device based on the tortoise shell that writes itself into the same position every time when it's delivered. The device is designed to deploy in the stomach, which is unique, and to inject a single microneedle spike that they call a millipost into the wall of the stomach. Several small devices are deployed at once, as you can see it from the figure on the bottom right, and a spring actuates the delivery of the millipost. My estimate is that each capsule contain up to 10 milligrams of active drug based on other prior systems that were based on single spike injection for sub-Q delivery. The mini capsules are excreted intact, and Novo is partnering with MIT on the development of this system. Another system out of the MIT group is the Lumi device, and it's actually fairly similar in its construct to the Lindra delivery system, where the device is folded up on itself inside a capsule. The system's based on microneedles and has the same limitations for drug loading as most do, up to about 1 milligram per microneedle dose. When the capsule shell dissolves, the device springs open, making the microneedles available to contact the inside of the gut for penetration of the drug. The publication and work in the reference was supported by NIH, and this program is also being developed in coordination with Novo. Progenity is a diagnostics company that recently got into the precision medicine business when they purchased the Medimetrics IntelliCAP device for sampling in the environment, as well as using it to deliver active drugs. The system is electronically controlled and can be delivered at any position within the gut, which is one of its distinctive differences from the other systems being discussed. So colon delivery, for example, is achievable. It contains a liquid reservoir of 0.3 milliliters, which would suggest that up to about 30 milligrams of a biologic drug can be delivered from a single device. Another operational tool is that it can be delivered, drug can be delivered continuously or in a pulsatile fashion, and this is all controlled remotely from a wireless system. In the past, this was used as a diagnostics tool for gamma scintigraphy and other mechanistic studies, as noted here, and there's a reference on the bottom which describes the system. The Entrega device recently had a patent published, and I was able to pull some information from there. They're using a drying agent to reduce the amount of GI fluid present when the formulation dissolves. This has the effect of maintaining a concentrated formulation at the gut wall for longer periods of time. They're also investigating superporous hydrogels that deploy and create pressure within the gut to hold the formulation against the wall of the gut. They can formulate product in a tablet or a capsule formulation, and they've reported several percent bioavailability in the patent. They are a PureTech company, and they are working in partnership with Eli Lilly on peptide delivery. Well, now let's move over to talk about the BaywindBIO Ventures JetCAP technology. This is a capsule-based device, also containing a liquid reservoir in the order of about 0.3 milliliters. And from a 100 mg per ml protein solution, this volume could deliver up to about 30 milligrams. The system's made of standard pharmaceutical components that are well known from a manufacturing perspective: a tablet, a capsule, a spring, a plunger. The capsule is made of cyclic olefin polymer, which is used in prefilled syringes for biologics formulations that are sensitive to glass. The actuation mechanism is an enteric-coated tablet, which when it dissolves, releases the spring to push a plunger down the reservoir. The capsules have holes or nozzles in the wall of the capsule at the end of the reservoir, and I'll show you some more detailed pictures. And we anticipate 30 to 50% bioavailability and are readying for dog PK studies. Here's a detailed description of how the system is working. Figure on the left is the finished capsule in its starting position as delivered to a patient. The blue is the fluid in the reservoir, and you can see an arrow pointing to nozzles on the bottom left of the screen, where these would be enteric-coated with the same enteric coating as the tablet. The plunger is represented in purple. The tablet is represented here in brown as an enteric coated for release in the upper part of the GI. And the tablet is holding back a medical grade steel spring, which is designed to push the plunger down the barrel. The middle panel shows the release of the spring after the tablet dissolves, pushing the plunger, and the spring expands to hit the plunger and force the fluid toward the nozzles. The fluid's forced out of the nozzle into a columnar stream of a given velocity depending on the nozzle diameter, the fluid viscosity, and a number of other parameters. The right panel shows the capsule in its fully emptied state, which will then be excreted in the whole capsule as shown. The fluid can be aqueous solution or non-aqueous solution and may contain soluble drug or a suspension as long as it fits through the orifices. Now let's talk about some of the development data and the origin of this device. The JetCAP was conceived from the concept of a prefilled syringe. And here you see in the picture an Instron device being used to test the pressure in a prefilled syringe. If you push hard enough on a prefilled syringe, you can create a nice columnar stream of fluid. And the initial work that was done here on the right shows the force required to penetrate chicken liver tissue, and the dye shows the impact points as pointed out in the bottom right of the screen. The literature has a few examples from needle-free injections that show the forces required to penetrate various tissues. As you imagine, the force required to penetrate the lining of the gut is much lower than that which is required to penetrate skin tissue to deliver drug to the subcutaneous space. It is also well known that the GI is highly vascularized, and an injection here delivers a large amount of drug into the systemic circulation. Ronnie has proven this point with their microneedle data. The next generation of the device was designed based on prefilled syringes that had holes drilled in the sides near the bottom of the barrel. Here you can see a fluid stream being created from a force delivered from a prefilled syringe. And as you can see the setup in the bottom left of this figure, we have the instron tester in an expanded view there with a prefilled syringe on the inside of the metal device there. You can't actually see the prefilled syringe, but you can see the stream of fluid, which has been darkened and is colored red there highlighted. The stream of fluid is on the order of 10 centimeters long before it begins to spread. So it is a quite narrow column of fluid. The upper right shows an expansion of the fluid to show how it is nicely ordered columnar stream. The bottom right shows the diameter of the stream is less than 1 millimeter from the hole in this prefilled syringe. And finally, it takes about 1 to 2 seconds to emit the delivered volume from the prefilled syringe. Obviously, this is dependent on the volume being emitted and the force being applied. The forces we demonstrated from this device have been used to design the spring needed for the capsule. Let's move on to that in the next figure here. Finally, on the right side of this slide, you can see the first prototype that was made from the device, which was made out of stainless steel with nozzles drilled into the sides of the reservoir. You can see in the picture on the right, the nozzles that have been drilled into this device. This has been tested in the same instron tester, and you can see that the plunger there was being used from the barrel of the prefilled syringe to test these. Final prototypes are being tested in dogs, and they will be composed of cyclic olefin polymer, as shown in the figure at the bottom of the page. The 0.3 milliliter reservoir is on the left, and the actuation end of the capsule is on the right. The device is approximately the size of a 000 capsule or a large amoxicillin pill. JetCAP is part of the internal innovation engine at BaywindBIO Ventures, where technologies are incubated using internal resources. Patents are filed and preclinical data is established prior to partnering with various funding sources, including investors and pharma and biotech companies. JetCAP is in the process of being readied for a dog PK study, and there has been significant pharma interest with some already in active discussions. You can see that we have a patent pending, as shown in the reference on the bottom of the screen. In terms of a comparison of these technologies, 2 of the systems are based on microneedles and 2 are based on delivering fluids from the device. Liquids can potentially deliver higher payloads, whereas needles are limited to smaller dose drugs because of the limitation of the amount of mass that can be put on a microneedle. The advantages of the microneedle is that they will only penetrate to a certain defined depth of the tissue, whereas a liquid stream will be harder to control in terms of penetration depth. The formulation options for fluid streams are broader, since solutions and suspensions can be delivered in both aqueous and non-aqueous vehicles, opening up a wide variety of formulations of biologics that might be conceived. Viscosity will surely play a role in the liquid delivery systems at higher doses and higher concentrations of biologics. There's a lot of interest in this space and much more data to be presented. Finally, I'd like to conclude by saying that converting biologics to oral products is a huge opportunity financially, and also there's a great patient need for these delivery systems to make more biologics drugs access to more people. There's also the potential to deliver topically to the gut from solution systems, and Progenity has shown some proof of concept data with an antibody delivered topically. In addition, the liquid systems may have the potential to deliver longer acting formulations, which could allow for ultra-long oral delivery of a week or a month. There are also vaccine and device applications which could be conceived of by delivering other kinds of agents to the GI tract. Thank you for your time and attention. I will take questions now. Thank you.